Good afternoon. This is Dr. John Bennett broadcasting from sunny Miami. Today in Larkin Hospital TV and the Neurosurgical channel, we once again have Richard Mendel MD, spinal surgeon from St. Petersburg, Florida, who's going to talk about stem cells today. And we're also joined by two distinguished foreign panelists. And Frederick, welcome back to the fold.
Hi guys, live from Belgium here. It's been a while, but I'm ready for another great hangout.
Well, it's good to have you back, Frederick. And my main man, Simon, in Japan.
Hi, thank you so much. It's always a pleasure to be here. I'm looking forward to this. I'm a medical student in Tokyo. Thank you so much.
Very good. Welcome, Simon. Okay, Richard, it's all yours.
Okay. What we'll do today is just kind of start a multi-series, multiple episode series on stem cells. And it's not going to be directly applicable to neurosurgery per se. We're going to just talk about where the science is now, and I'm going to try to give you a brief introduction, but enough background to understand what is worth talking about and what is, not really valid at all. So the brief introduction. What we're going to talk about over the next few weeks is the science and the medicine of stem cells and the reasonable expectations over the next few years on where things are going and what is possible now and what is just complete science fiction. So hopefully you walk away with this, with some of the fundamental knowledge that has come out of the research that's really been going on since about 1981, when the first notion of stem cells in mice and the development of knockout mice occurred, and that was 1981, and that work plus some of the manners in which to isolate and manipulate stem cells got the research from 81 as well as researchers from the University of Utah and the University of Wisconsin Nobel Prizes in 2007. So we'll kind of go over what's been done so far, but we're not going to talk about ethics, we're not going to talk about laws or politics. There's many good publications on that, and I'm not even going to touch on it. We're just going to talk about the science and give you a working knowledge so that when you look through a paper, you understand things enough to know what is valid and what's not valid. So to start out, there's two criteria for a stem cell. Okay, it's got to be able to reproduce itself, and it's got to be able to generate the offspring of different functional cell types. The notion is that the stem cell is like this omnipotent, universally applicable cell that can generate almost every cell in the body. And that is somewhat of a myth right now. Okay, so we'll get to that in a few minutes, but you have to think of the cell as a structural unit. So when I was taking biology just as a high school student, you would often hear about the nucleus being the brain of the cell, and when you think about it, in some ways it made sense, but the nucleus really just contains all the genetic material, the DNA, as well as genetic components and genes that dictate what the rest of the cell is going to do. So the cytoplasm is where your proteins and all sorts of molecules are that do the mechanical and biomechanical tasks. So when you think about gene expression, that describes the production of proteins from the active genes, right? And we often, even doctors and scientists will refer to genes as genes, and you think, well, a gene will just produce X or Y. And you have to remember that the gene has variants, and the variants of those genes are the alleles. So we really should be referring to a gene as its allele type, not really as its, quote, gene type. And so the genome itself is, of course, the set of all genes present in the cell nucleus, even though the differentiated cells are going to have a tremendous amount of these genes completely turned off. Okay? So now this is where things start to get kind of complicated. We talk about cells as the structural unit of animals and plants. But basically you can break up cell lines in your thinking into two categories, undifferentiated and differentiated, okay? And if you look at the last statement, it says stem cells can only be defined by their behavior. So a cell really is only defined by what proteins it produces. And so when you look at an undifferentiated cell, when you look at it, may be a kind of bland, not interesting looking cell under the microscope. And you may very well think it's undifferentiated, but you won't know that until you know what that cell with its genes are set to produce. So until you can look at its production, you don't really know if it's an undifferentiated cell or not, because as you're going to see, there are many, many cells that appear undifferentiated but have some specialization or differentiation that only gets recognized by things like we talked about weeks ago, which is the CRISPR-Cas9. Okay? So if you think about a couple weeks ago, do you remember when we talked about neuronal? You don't really know if it's an undifferentiated cell or not, because actually,
Neurodifferentiation? Oh, I'm sorry, there's an echo? Okay.
Have some specialization or differentiation. Yeah, I'm not sure what's going on. Yeah, that was better. It stopped now. Can you hear me?
Yeah, that's from me, Richard. I'll turn myself off here.
Okay, that's all right. So anyway, you're not going to know really whether something's differentiated or undifferentiated just by appearance, okay? Now, when you think about differentiated cells, usually they're going to have a typical characteristic. It seems like every medical student you ever meet, every doctor you ever meet, after they've taken pathology, no matter how brain dead they become, the minute you flash up a hepatocyte, they recognize it. Like, I don't know what it is. And neurons are pretty easy to recognize. These are examples of differentiated cell lines, hepatocytes, cardiomyocytes, neurons. But when you get back, these are clearly structurally different than undifferentiated cells. But because it's the protein that the gene produces that defines the type of cell it is, it just may not be all that interesting looking. It may not look like a neuron, but it may be what's called a progenitor cell to a neural cell type, or a cardiac cell type, or a hepatocyte. Those are all kind of interesting looking structures. So let's get back. I want to skip back here and reinforce this. The archetypical stem cell is the embryonic stem cell, and it's a mythical human-created artifact rather than an entity that we can really define in nature. It's kind of like Zeno's paradox, where you shoot the arrow at the target or the bullet at the target, and if you take an infinite number of photographs, you can kind of make the argument that there's just never one moment when it actually permeates the target, you know? So this is from Aristotle, the syllogism, right? So syllogism is where you have this major premise and a minor premise. So you say undifferentiated cells may appear generic under the microscope, but it's the protein that its active genes produce. Therefore, you know, an undifferentiated cell, most undifferentiated cells are specialized in some way, often in a way that restricts it from being another type of cell. Okay, so we say that all stem cells are of an undifferentiated type, but not all undifferentiated cells are stem cells. Does that make sense? Stem cells are undifferentiated cells, but not all undifferentiated cells are stem cells. That becomes important later on because, you know, there's many criteria of what a stem cell actually is. So they need to meet those two criteria that we talked about. It has to be able to reproduce itself and also reproduce other types of cells in the organism that it belongs to. So let's get back to, I want to go to renewable tissues. Okay. All right, so now we're going to talk about renewable tissue. And a good example of renewable tissue is your skin or your intestinal lining or hematopoietic cells in the blood. All right, but for this purpose, we'll just talk about skin cells. So, you know, we have an epidermis that's essentially, you know, comes from keratinocytes, and you know, once the cells die on the epidermis, they just slough off. But we're constantly producing new cells, and you know, the skin reproduces itself incredibly fast. So does the hematopoietic system or the GI system. So with the skin, you have this outer layer of the epidermis. It has this keratinocytes, and they're constantly being worn away. But they're constantly also being reproduced in the basal layer. So what happens is we have a basal layer that contains a type of stem cell that can't produce every type of cell, but it can produce every type of cell that's going to be part of the epidermis. So 50% of those cells in the basal layer will remain there to amplify the production of more cells that would eventually travel through the entire skin layers to the epidermis, and as it travels, it will change the activity of the genes in it and change the output of the active genes to produce more and more keratin as it gets closer and closer to the surface. So this is kind of a diagram of a renewable tissue. This is the stem cell, and in a way, it's probably not the idyllic embryonic stem cell that can perform that can produce neurons, cardiomyocytes, hepatocytes, but it very well may be able to produce. It can produce every layer, say, of the skin or the dermis. So there's a basal layer that contains stem cells, and then it has what are called, instead of progenitor cells, it's called tissue amplifying cells that really just step up the production of all these differentiated cells that end up on your skin or in your GI tract or in your bloodstream. So these are what you call tissue-specific stem cells. But you notice that there is a component of this cell that's really not like the embryonic stem cell. It can produce its own specific tissue, but no others. So right there, you've introduced a degree of specialization. Let's see. So as you know, that I've already mentioned that, the degree of specialization. So now, as I mentioned, the most famous cell of all is that embryonic stem cell. And really, they don't exist in nature right now. We have isolated what are likely to be likely to meet our criteria of what an embryonic stem cell is, but they're not in vivo, they're in vitro, which is Latin for in glass. You know, they are cells that are manipulated and put in a growth medium so that they retain their pluripotency, their ability to develop into other types of cells for a short period of time. But they don't stay as embryonic stem cells for long. So the idea is that they kind of become a stem cell and the way in which they're limited is that they can't produce tissues outside of their own type. But for that, they're good at producing these progenitor cells, which provide a renewable source. So you have to be cognizant here of whether, when you're reading about something, whether it's an in vitro, meaning in glass, cell culture, or whether it's the notion of a pluripotent stem cell that can divide into all kinds of tissue types. Now, recently, that 2007 Nobel Prize from the professors at Utah and Wisconsin was really granted on the ability of them to induce a pluripotent stem cell that could divide into other tissue types. But that's really quite far away right now. This is what the problems in stem cell therapy are right now. Virtually every important cell type in the body does not grow during adult life. Okay? So when you think about the cells of an adult human, there's an estimate that there's about 210 different types of cells in humans. But I don't really know how accurate that is, because if you remember, weeks and weeks ago, we talked about Camille Golgi and the staining of neurons, and this was work done in Spain 200 years ago. And at the time, the thought was that there were 5 different types of cells in the retina of the eye. But I told you in that talk that it turns out, and that's what I was taught in 1989 when I graduated from medical school, there's actually 65 different types of cells in the retina of the eye. So I don't know whether the 210 takes those 65 into account or not. But you know, that was all from the work of Cajal, Raymond Cajal, Santiago Ramon Cajal, and Golgi 200 years back. So there may be quite a bit more in the way of the number of cells in our body, the number of types, because, as I said, we're not recognizing what to all outward appearances, look like undifferentiated cells. So the really useful types of cells that we need, like hepatocytes for the liver, or the beta cell, the islet cells of Langerhans in the pancreas, those, they don't divide in tissue cultures. If they, divided, they would lose their functionability pretty quickly. So today, most of the current therapy that goes on has nothing to do with amplification of cells. And what we do is almost all blood-based, hemopoietically based, in that we graft cells from one person to another, and the significant application of that is things like bone marrow transplants. That's the most important kind of current stem cell therapy. We're not able to up and down regulate, amplify different tissues. And so here are the remaining difficulties, okay? The first difficulty is finding a stem cell that that has a renewable source, and you can make enough cells to reproduce the function of an organ. But the other problem is immunity, and this is a two-edged sword. With immunity, our T cells are really efficacious. T cell lymphocytes of knowing what are, quote, not self cells. And so the immune system is great at finding cells that aren't intrinsic, that are foreign to us, and killing them. And that's done with cytokines, and interferons are just a type of cytokine. And interferons from all the oncologic applications. And then there's also the HLA system, which is the human leukocyte antigens. And those are the glycoproteins that are on blood cells. And it has a myriad diversity of permutations, really. And so you really would need to very fine-tune the immune system, or get perfect matches for people, and that's not realistic. And the last issue in immunity is the nightmare of immunosuppressive drugs for people that are getting transplants. People that even get a common transplant like a kidney, they're really impaired by the amount of immunosuppressive drugs that they have to take. So remember now the difficulty so far. The first heading is a source, or a renewable source of cells that can be exploited. Number two is that you have to impair the immune system or outregulate it. And number three is delivery of drugs. Okay, the delivery system of a particular thing that you want to graft is not well thought out yet. So if you think back to the 1980s, even the early 90s, you heard about a group of neurosurgeons in Mexico that were implanting cells into the substantia nigra and the basal ganglia of Parkinson's patients, trying to improve their function. And that's not, that's fraught with all kinds of difficulties, and we're only now getting to get a really good grip on the immune system itself, let alone neuroimmunology. So the holy grail for stem cell transplantation is really going to be producing a cell, tissue, or organ that doesn't need immunosuppression. And that is the lead into what we're going to talk about next time, which is personalized pluripotent stem cells. Because taking an individual genetic makeup and looking at their alleles, not just generalized gene production, but the alleles that they have, and trying to taper something for them in the future. Having said all that, the last slide is kind of important. It's also kind of sad. You know, we have what goes on now in medicine is aspirational stem cell therapy, and a lot of bench scientists just talk about stem cell tourism because there's a lot of money involved in this. If you remember, a very, very well-known quarterback in Indianapolis went outside the country to have stem cell therapy to fix the cervical disc he had herniated in his neck. Now, he apparently went to France, and he had blood drawn out of an IV line and then re-injected. I don't know if it was the same day or later in the day, and then had his anterior cervical discectomy done. And unfortunately, that failed to fuse, which is a very common problem with the operation, an anterior cervical discectomy. But certainly, the withdrawal of blood from his vein and re-injection later that day or at the same time really was wishful or magical. This quarterback could have gone to one of the best spine surgeons in the country that lives right in Indianapolis, but somehow, you know, because of people he trusted, you know, he went to some place that promised the sizzle of stem cell therapy or stem cell tourism. So when you watch commercials on TV and you see a Neutrogena product talking about messenger RNA and revitalizing skin, I mean, it's kind of absolutely ridiculous. The keratin in the top layer of skin is dead anyway, so you're not bringing that back to life. And so media and commercials are really ridiculous when you understand where we are in terms of stem cell therapy now. So I hope that gives you just a general introduction. What you know right now is a stem cell has to be able to reproduce itself and has to be able to generate numerous of all the progenitor cells in its particular type of tissue. But the iconic embryonic stem cell, we've never seen it in nature. We talk about it and we put it in vitro in glass, and we manipulate something to the point where it can diverge in a huge algorithm to all kinds of different cells, but that's not something that's been discovered in nature yet. It's just an idea we have. So are there any questions right now? I mean, I think it's going to come together a lot more as we start talking about specific topics, but you have to take these aspirational stem cell therapy and this stem cell tourism nonsense with a grain of salt, because there's a lot of money involved and there's very little science. And, you know, unfortunately, a lot of medicine, you know, double-blinded controlled studies weren't even invented to the 1940s. So you can fool yourself into thinking you're doing something that yields great results, but you know, there's not a scientific basis for it. So you have to be pretty stringent about how you're going to talk about the stem cells as we proceed. But you understand now we're not going to talk about he has this gene or that gene. We're going to talk about the alleles of the gene rather than you know, just because somebody has an impaired gene doesn't mean it's going to be a problem. You know, we know that with certain types of dementia, the haplotypes and the different types of genes or the alleles that they have a high predictability for things like Alzheimer's disease and dementias. So.
Well, you know, Richard, thank you very much for a very clear presentation with great slides. And this is one area, as you know, that needs education, like you mentioned at the end, because there's just too many people being defrauded, going overseas, believing the hype, and we really need people like you educating other people about what's really going on. And we hope to use Hangouts to try to help to educate people. One question, Richard. Now when they say, you talk about embryonic stem cells, are those stem cells that are in the placenta? Because I've heard a lot about controversy about placenta stem cells, and there's a lot of controversy about, you know, those kind of things.
Well, yeah, that's a great question, and you know, I think the people, I think in Britain they do a lot better job with the vocabulary they use. See, here we talk about embryos, and you're right. I mean, that's what we're talking about. With human stem cells, it's better to think of what the embryo is, because the embryo that we talk about is really better called a conceptus, because once the sperm and egg come together, not only does it generate the portions of the body on the baby, but it also generates a portion of the placenta. So both the mother generates a portion of the placenta, and so does the conceptus, or what we call the embryo.
Mm-hmm.
What we're really talking about is an embryo and a piece of placenta. That's the conceptus that comes out of the union of the sperm and the egg.
Mm-hmm.
So,
Okay.
you know, probably the right way to talk about it is to say it's a conceptus, but we kind of all use the word embryo now. It's kind of it's not really right, but that's what we talk about. So yeah, the portions of the placenta that are maternal versus embryonic do fall into a gray zone, and it's important to know that. But I'm not going to talk about, like I told you, the ethics, laws, or anything. I'm just going to mention the science of it. And the science is that when the sperm and egg come together and they form that trophoblast, which is the layer around the thing we call an embryo, that embryo also gets that embryo also generates a portion of the placenta. So that's why it's probably right to call it a conceptus, but nobody else does except the people in Britain are just more exact, which is good. It's clear.
Okay. Okay, Simon, do you have a question?
Oh, yes, please. Thank you very much, Dr. Mendel. I always love to listen to your basic science discussions, and I'm fascinated. I just finished a hematology, oncology, immunology block, 6 weeks, and I took a 100-question exam a couple of days ago. But, you know, looking at your presentation, I realized they still have a lot of questions, and there were things I was not recognizing. And when I talk to working physicians and I talk to them about what I'm studying, they say, boy, I don't remember any of that. And I say, you're kidding. You know? And I said, well, it's so important.
Yeah, well, it's important really to the basic scientists, right? Mm-hmm. Because, you know, if you're a clinician, you're worried about the patient. And so mm-hmm. you get these different, and this is very important culturally, right? If you're a basic scientist and you do something like inject some bone marrow into a kidney and the function of the kidney improves 2%, mm-hmm. and but you know that every one of those bone marrow cells is dead an hour later, you say, well, we don't need to look at that any further. But the clinician says, hey, that patient got 2% better. I don't care what the science is. As long as they got better, I'm happy. And, mm-hmm. and it's, you know, that's an extreme view, you know, something, you know, I just made up. But it's not, it's not the right view on either side. And, you know, I think the thing that's really happened over the last 20 years that's making things much more interesting is that two different groups of people are coming together. You have biological scientists or doctors and basic researchers, right? And when they take something from life in vivo, in nature, and put it in glass, in vitro, right? The biologic scientist thinks of that as an artifact, and it's a negative thing, because what they want to do is understand nature. They don't want to understand, they don't want to look at contaminated contaminated instances of nature. They want to look at actual natural processes, right? But the bioengineer, who are getting more and more involved, they now want to look at ways to make products, useful products for people, right? So they look at that artifact like a giant plus, because you've taken something from nature in vivo, and now it's put in glass in vitro, and the cell adapts some way, or it's manipulated some way, and then the engineer says, well, you know, we can manipulate X, Y, and Z and produce something that's, so you're getting a new look at things by the engineer, who just come from a different culture. And the scientists made it possible for these engineers to get into bioengineering, and that was a book by Carlson called Biology is Technology. It's about 10 years old now, but you know, it heralded the engineers and people to start manipulating biologic systems rather than mechanical systems. So yeah, you're right. Like the hematologists you work with, they may know an infinite amount about the cells that they can manipulate, but the things that they can't manipulate, or that just that haven't presented themselves yet, aren't as important to them.
That's what I'm sorry. Yeah, they aren't as important to them. Yeah, and that's the point. Yeah,
you're exactly right. But you have to remember that over the last 2 or 3 years, the whole world has changed in immunology.
Yeah.
Now we're better at manipulating the immune system, and that ability has actually become like another tool for oncologists. They have, you know,
do you think there's a Higgs boson of medicine on the horizon?
I don't know. I'm certainly not going to live long enough to see it, but yeah, there's probably an infinite direction down and an infinite direction up, you know.
We've observed, we haven't explained, and I would like to see more explanations.
And now we're in an interesting point, because we're at a point now where we're observing things and we're not even able to see that they're specialized. You have to actually see what they produce to know that they're specialized. So yeah, it's very interesting now, because we're just going into, I mean, these worlds were unimaginable, 200 years ago. Yeah, it's a whole nother world now. It's getting really interesting.
Yeah, we hope to go to these places with Hangouts.
Yeah.
But Richard, I'd like to, I don't know if you met Bernardo de Andrada, from a spinal neurosurgeon from Rio. Have you met Bernardo? Hello, Bernardo. Welcome, Bernardo. I can't hear you, Bernardo. I think you're muted.
That's great.
Oh, hello, Bernardo. Oh no. Oh boy. Oh. Well, anyways, Bernardo. I think he's come to some of your hangouts before, but yeah, it's just a fascinating area, and we can go in many places. And I guess you're going to have a few more hangouts about stem cells, right, Richard?
Yeah, I mean, it's probably going to be 4 or 5 of them, I think.
Oh, good.
To cover it, because
yeah, like I say, this is one area of medicine that really needs education.
Well, we're going to come back to a loop, because if you see the CRISPR-Cas9 system, was the way to retrace genetics, because you could use these palindromic sequences and it would tell you what the gene made, and then that way you could characterize the gene, and also that reflects back on when we talked about the brain's identity crisis, that article that appeared in Science a while ago. So yeah, we're going to be able to backtrack and really date things a lot better in nature that have come before.
Well, you just hear of so many disastrous cases where people go to Latin countries and Korea and China thinking that they'll cure arthritis, or
yeah, well,
for $30,000, $40,000, $50,000. It's a shame so many people are scammed in the stem cell area.
Yeah, well, you have to buyer beware. If you don't understand the science, it's probably not, it's probably not real.
That's very good. Okay, we'll end this broadcast, and we'll just hang around, see if you can get to talk to Bernardo. Thank you, Richard. And thank you, Simon.